So welcome everyone. We are very excited to be here tonight and to be hosting our two speakers. And we're very excited to have you all here. So thank you all for joining and for tuning in to our Science Cafe. My name is Alicia Kojohari, and I'm a postdoc here at the University of Massachusetts Chan Medical School in Worcester, and I will be your host tonight. And joining me are Priti Thakur and Sneha Shah, who are also researchers here at UMass Chan, and they will be helping me with the Q&A session. So our topic tonight is piRNAs, and we're very excited for it because piRNAs are a type of small RNAs which have originally been implicated as being very important in maintenance of male fertility. And more recently, they have also been shown to be implicated in some other processes such as virus defense and cancer. But in today's Science Cafe, our two speakers will be talking about piRNAs and how they're made in cells and their role specifically in fertility. And before we get started with introducing our speakers, I first wanted to quickly remind you that the views in this presentation and the speakers' presentations are only those of the presenters and not necessarily those of the UMass Chan Medical School. And also our Science Cafes are recorded and will be uploaded on our YouTube channel, which you can follow here. You can see it on the screen, and it's also in the chat box. So if anyone wants to share the seminar with your friends and colleagues to view later, please do so. So please follow our social media channels as well. The links are again in the chat, so you can stay up to date with our upcoming events. Also, if you have questions during the presentation for the speakers, feel free to raise your hand and we will call on you and you can unmute and ask the question yourself, or you can type the question in the chat box and we will ask it on your behalf during the Q&A session. So now we will go ahead and get started. Our first speaker tonight is Milky Abajorga. Milky is a fourth-year graduate student studying piRNAs in the labs of Doctors Jeremy Luban and Bill Theurkauf here at the University of Massachusetts Chan Medical School. Besides science, Milky enjoys napping, crocheting, and hiking. So Milky, welcome. Thank you so much for being here tonight, and you have the mic. So I will unshare now if I can, stop sharing. Okay, there you go. Perfect.
Okay. All right. The title of my talk is Development of piRNA Response Protecting the Germline from Retroviruses. Now, before I go into the nitty-gritty part of my talk, I'm just going to provide a brief introduction so that it'll be
recording in progress.
So that it'll be easier to understand the rest of my talk. So in a cell, there is a compartment known as the nucleus right here, and the region outside the nucleus is known as the cytoplasm. So the nucleus is where all of our DNA, the DNA is located, and the DNA is made up of two strands right here. Like there's the top strand and the bottom strand, and they're connected to each other using intermolecular forces. And DNA itself, each strand itself is made up of monomers or molecules known as nucleotides. Now, what happens, so all the DNA, I just want to point out that all the DNA in the nucleus is known as the genome. All the DNA we have is known as the genome. Now, what happens is the DNA goes through a process known as transcription, and what happens is the information encoded in the DNA is transferred into RNA. This RNA is also referred to as transcript. So then what happens is the RNA moves to the cytoplasm, the area outside the nucleus, and it is made into a protein by big molecules known as ribosomes. And so I just want to point out that proteins are macromolecules that perform specific functions. Now, in the genome, there are these small pieces, there are these specific pieces of DNA known as transposable elements or transposons. Now, what transposons can do is that they can move, in that, for example, this transposon found within a specific place in the genome can move to another part of the genome, and this process of the genome, and Because of that, transposons are also known as jumping genes. Now, transposons can threaten the stability of the genome. For example, a transposable element or a transposon can insert itself into an important gene and hence, for example, impair the function of the protein that is made from this gene. What is interesting is that transposable elements are actually very common. They're found in humans, mice, and fruit flies, as well as worms. And in humans, they make up almost 50% of the genome. In mice, they can make up almost 38% of our genome. Now, since they threaten the genome, there has to be a way to stop them. And one method are PIWI interacting RNAs. So if this is a transposable element right here, it can threaten the genome, but then PIWI interacting RNAs or piRNAs repress transposable elements and help maintain the genome. So what are PIWI interacting RNAs? So PIWIs are single-stranded RNA, but they're smaller than a normal transcript. They're about 21 to 35 nucleotides long, and their length depends on the type of PIWI protein, the organism, and the organ itself. And these piRNAs are loaded specifically on PIWI proteins, hence the name PIWI interacting RNAs. In vertebrates, they're found predominantly in the testes and in the ovaries, so in maturing sperm and eggs or oocytes. But then you can also find them in somatic tissues, that is cells that are not sperm or egg, in other organisms such as fruit flies, for example. So one of their most important and actually well-characterized function is that it represses transposable elements. The way it does it is there are two mechanisms. The first one being, if this is a PIWI that has been loaded with a piRNA, it can bind to, for example, a transposon transcript, and the PIWI protein consequently cleaves the transposon transcript. Another way it does is the PIWI protein, which has been loaded with a piRNA, moves into the nucleus, and then it moves to where the transposable gene is located. And what it does, it recruits several proteins, including histone and DNA methyltransferases, and it causes chemical modifications on the DNA to prevent the transcription itself of the transposable element. So, but the question is, how is it that piRNAs are able to recognize between the good and the bad? So if this is a transposable element that is being made or that is being transcribed, and there's also a host gene that's being made and transcribed, why is it that the host gene transcript is loaded onto ribosomes, and why is it that a transposable element transcript is loaded onto a PIWI protein? Because both of them, both a transposon transcript and a host gene transcript have nucleotides. They aren't different. They're not made of different things. So how is it that the host, the cell, can differentiate between these two? Another question we have is, for example, if there's a retrovirus and it has integrated into the germline specifically, so into the sperm or egg, and it becomes a permanent part of the genome, in that it can be inherited from one generation to another, to the next, because it is found in the germline. So if this happens, how is it that the piRNA can recognize this new insertion as a threat and repress it? So in order to answer these two questions, we're testing these in 3 different organisms, koalas, mice, and fruit flies. But for this talk, I'm just going to talk about what we've been doing with koalas. So koalas right now are being infected with a retrovirus known as CORV-A. And CORV-A is currently spreading from North to South Australia. So if we look at this right here, if we look at animals, koalas specifically from the northern part of Australia, almost all animals have CORV-A in their germlines. I should make a big distinction. So CORV-A can be transmitted in somatic tissues, and that it can be transmitted from koala to koala. But the difference is endogenous CORV-A is the one that's being transmitted from one generation to the next. And I just want to make that distinction. So what we're looking at right now is endogenous spread of CORV-A. So from one generation to the next, CORV-A is found in sperm and egg. So, but then when we move from the north to the south, fewer and fewer koalas have CORV-A in their germline. So the approach we took is we got tissues from our collaborators in Australia from 3 different regions: Sunshine Coast, Gold Coast, and Adelaide. After we got the tissues, we extracted the DNA and RNA and did sequencing. So I just want to point out that I'm just going to focus on Sunshine Coast and Gold Coast specifically. So if we look at animals from Gold Coast as well as Sunshine Coast, so the x-axis right here is the length of CORV-A, and the y-axis is the depth, which is used as a proxy for expression level. So the higher it is, there's more expression. So all the animals from Gold Coast and Sunshine Coast have CORV-A in their germline. And when we did our DNA sequencing, we know that they have about 70 copies each. So, but then if we look at the RNA, I should repeat this. So DNA is going to be transcribed into RNA. So right now we're looking at the RNA. If we look at the animals from Sunshine Coast, we see almost no expression. The expression level is pretty low. But then if we look at the animals from Gold Coast, you see areas of higher expression right here. So what's the difference between these 2 animals? Because both the animals in Sunshine Coast and Gold Coast both have about 70 copies, but there's lower expression for the animals in Sunshine Coast. If we look at the piRNAs, the ratio of the sense to antisense piRNAs is different. So animals from Sunshine Coast, the ratio of antisense, so the top strand is sense and the bottom piRNA. So the ones that are transcribed from the top are sense and the bottom is antisense. So they're both sense at the top and antisense piRNAs right here. However, if we look at animals from Gold Coast, most of them are coming from the top strand. So what is happening here? So what I want to show is that as animals are moving from the top to the bottom, there's endogenization of CORV-A, but then there seems to be some sort of adaptation. But what is exactly this adaptation? So if we look at animals from the Gold Coast, if this is CORV-A, what we think is happening is this CORV-A is just being chopped up into piRNAs. And that's how the cell is able to reduce the amount of CORV-A expression. However, if we move the animals to the Sunshine Coast, CORV-A is being transcribed from the top and the bottom, and the top strand is made into sense piRNAs, and the bottom is made into antisense piRNAs. And the reason that is important is because a pili protein loaded with an antisense transcript can complementary bind to the CORV-A transcript, so the one that makes the virus. And then that could cause the degradation of the CORV-A transcripts. So that's the major, what we're hypothesizing is that this is happening right now. And with that, I would like to thank members of my lab, and I'm happy to take any questions.
That was great. Thank you so much, Milky. So if anyone has questions, please type them in the chat box. So maybe I'll get started with a question. So you mentioned the sense and antisense piRNAs. Are they made from sense and antisense viral transcripts? And do sense and antisense viral transcripts both have a function? And did you look at the expression of viral? I know you showed the expression of transcripts for the virus, but was that sense or antisense transcripts?
So when we're looking, I'm sorry, I'll share my screen again. So when we do the RNA sequencing, we're looking for both anything that will align to correlate, whether that be sense or antisense. And so when we're looking at this, there seems to be nothing either from the top or the bottom strand. So to be honest, what I think is happening is there's an epigenetic change made with CORV-A, and so there's no, and also any little, besides the epigenetic conversion that would repress the transcription, any remaining CORV-A transcript that might be transcribed is being made into piRNAs, and they're being chopped up. So we don't see any antisense transcript. And here there is some antisense right here, which we do see here. There's some antisense piRNAs, but here's the thing: the level of antisense right here is no different than the one here. The ratio is the one that's different. This ones have these ones, the animals from Gold Coast, like, have a lot of sense piRNAs relative to the antisense piRNAs. So the amount, I think the absolute value of the antisense piRNAs is not any different. So I think there's some sort of epigenetic change that has happened. I don't know if that answers your question.
Yes, it does. And I have a follow-up question, actually. So do the sense and antisense piRNAs have to be present to suppress transcription, or not necessarily?
Yes, so the suppression process is dependent on transcription. So because the PV protein, this is an antisense antisense piRNA loaded onto a PV protein, it has to recognize that transcript first and then repress it. So yes, it had or at least the beginning of the epigenetic change requires transcription.
That's great. Thank you, Milky. That was a very interesting talk. I had a small question. So what does CORV-A do, and how does this really help the koalas? So the idea is probably that it does not spread from generation to generation. And does that have any effect on the tissues itself?
So CORV-A, if it's moving exogenously just from koala to koala, it has been shown to cause leukemia and lymphoma and myeloma. However, I think the thing is, if koalas don't have piRNAs, likely they're going to be sterile. Just because there are no CORV-A piRNAs, I don't think that necessarily would cause sterility. But I guess it could mess up the genome if there's a chance that it inserts itself into an important region of the genome. But I don't think purely the absence of CORV-A piRNAs would cause sterility.
Interesting. So is this a very common mechanism for any retrovirus that gets inserted into the genome? Is this how the body fights, or are there any other ways as well?
Here's the thing. This is the CORV-A is the only, I think maybe there's one in chicken, but most endogenous retroviruses joined genomes millions and millions years ago. There's really, I think CORV-A is the one chance we have to see the endogenization going on right now, but otherwise most of them happened a long time ago that we can't really actually see it live, if that makes sense. So I cannot really fully answer your question.
But that's an interesting thought. Thank you.
I also have a next question.
Preeti, we cannot hear you very well. No, sorry. I'm just guessing you're asking if we can hear you. No, we can't.
Can you guys hear me now?
Perfect, yes.
Okay.
So Milky, I have a question. Does piRNAs are always specific to your transposable elements, like whatever we have in the genome?
Right.
Might be like always specific for the
Right.
virus.
So yes, well, maybe this is not the most updated slide, but I can use it. So what we think is happening is, so piRNAs, or the piRNA system, is an adaptive response because in the sense that it's specific. However, we think that there is an innate response too, which is why we think, so it's the thing we're seeing with the animals from the Gold Coast. We're just, it's recognizing something about CORV-A as bad, and it's making it into piRNA. So this is not necessarily specific, it's just the reduction of the CORV-A transcript just by degrading it into piRNAs, and this is an innate response. But you're right, and generally for established piRNAs, it's specific. Here's the thing though, at the same time, I should say that the PIWI protein, like chemically, it's made such that it can tolerate mutations and stuff. So if there's a new retroviral insertion and it's closely related to an endogenous retrovirus that's already there, then there are possibly already piRNAs that can repress this new retrotransposon. It can tolerate some changes, which is why, again, they're important. But in general, you're right, they are, so each type of piRNA is specific to
the transposon elements. Okay, thank you so much.
Yeah.
Okay, do we have any more questions? And feel free again to raise your hand and unmute yourself if you would like to, or just unmute yourself and ask the question if you would like. Or if you do come up with another question, when we move on to the next talk, please feel free to type it up in the chat box and we will definitely ask it after the next talk. So if there is no further questions, I will share my screen again. Okay, can everyone see my slides?
Yes.
Yes, okay, good. Thank you. So thank you, Milky, again. That was an amazing talk, and thank you everyone for the questions. So our second speaker tonight is Dr. Maya Spichal. Maya is originally from Germany, and she earned her PhD from France. She is studying piRNAs as part of her postdoctoral work in the lab of Dr. Craig Mello here at UMass Chan Medical School. And in her free time, Maya enjoys going to horse shows and exploring new restaurants in Boston. Maya, thank you for joining us, and the floor is yours.
Yeah, thank you everybody for joining tonight. Can you hear me all right? Okay. So the presentation that I want to give you today is called Understanding Fertility, piRNAs in the Spotlight. So here I'm actually showing you a picture of my family. And so I want to talk about the germline, which is actually the sperm and the egg in each animal, and which we consider as immortal. And why do we consider the germline as immortal? It's because the parents can give rise to children, which in this case, here's my sister with her husband, and then my sister and her husband had also some children, which would then be considered the second generation, or as we scientists say, F2. And so I'm sure that my sister's children will also be able to have children one day, and this is going to be the same in every generation. So here in my lab, we studied worms, which are also called C. elegans as a species. And in wild type worms, which are worms that are found in the wild, we have more or less the same phenomenon. So if we put 6 worms on a plate where they have food, they will reproduce, and then in the next generation, we can again put 6 worms on a plate where they eat food and can reproduce. And this can go on forever, and we will always have worms that reproduce. However, we found a special mutant in the lab that we called a Mortal Germline mutant. And what is special about this type of mutant is that the worms at the beginning could reproduce similar as the wild type worms, and then over the generations, their brood size would drop. So less and less worms were reproduced until eventually they would become 100% sterile, meaning that none of the worms were able to reproduce any longer. And so we were studying this phenomenon, and we were wondering what is the cause of sterility in mortal germline mutants. And so in my talk, we will discuss different causes of this sterility. And so we already know that this type of mutant is caused by the loss of piRNAs. And so Milky already introduced piRNAs, and so you already know what piRNAs are. So they are these really small RNA molecules, and in fact, piRNA loss leads to infertility in a variety of organisms, and it is also a common factor of infertile sperm in men. So in fact, 10% of men are affected by various degrees of infertility, and mutation in PV, which is the protein that protects piRNAs, is one common factor for their loss. So as Milky already pointed out, piRNAs occur in flies, mice, and people. And also, we want to go through the basic structure of life, which Milky already also pointed out. So, but just to go through it again, DNA, if you're not familiar with this, we can basically see it as a basic instruction manual. And it's kind of the architectural plan of a house where we know how to build it. And then we have the RNA molecule, which is kind of a specific information to build one protein. And each RNA molecule that is made from DNA can also be called a transcript. And to go back to our architectural plan, the RNA molecule would then be in comparison just a node in how to build one specific part of this house. And in this case, our RNA molecule wants to make a protein. And if by using the same analogy again, then this node would encode for, like, for example, like a brick of the house to build it. And yeah, so proteins are important and can fulfill a lot of different functions. And in this case, there's one special protein that is called a PV protein that Milky already mentioned. And PV proteins bind to piRNAs, and so they protect them and they guide them to their specific RNA transcripts. And so the function of piRNAs, there's basically 2 different types of functions, which we already learned in the previous talk, but just to make you more familiar with it. So the first function is transcript sorting. Binding of piRNAs to specific transcripts can allow the transcript to be translated into a protein. And also, a second function of piRNAs is transcript degradation, for example, transposons and virus RNA, as we also learned in the previous talk. So these different functions are very important. And so where, and also another notice that piRNAs are obviously much smaller than the RNA transcripts that make the protein. And that's where we come to our poll question. So I want to ask you, how much shorter are piRNAs compared to the average RNA transcripts? So please type in the answer, what you think. Is it A, 10 times, B, 100 times, C, 500 times, D, 1,000 times, or E, 10,000 times? So I'm curious what you would think.
Can everyone see the poll on their screen? I just want to make sure that people can see it.
I mean, if you type in the wrong answer, it's not a problem. Yeah.
Okay, now we see the poll. Okay, great. Thank you.
Oh, like more.
Okay, now people are voting, so we'll give them another 20 seconds.
Okay.
All right, anyone else? Feel free to either choose an option or type it in the chat box, and we'll end the poll in 5 seconds. So, all right. Okay, and then can you see the results?
Yeah.
Okay, great.
So yeah. So in fact, 2 of you were correct, and one piRNAs are about 100 times smaller than the average RNA transcript. So yeah, can I advance presentation?
I think you can click the arrow at the bottom left, maybe. Okay, there you go.
Okay. All right. And yeah, and so where are piRNAs located? So as we already know, like a cell is a building block of all living organisms. And so the cell is basically a container for the nucleus, which contains the DNA. And piRNAs are located right at the nuclear envelope of the nucleus. And so we want to investigate that a little bit further. And so when we look at this very closely, we can see that they are actually partially located in this structure called a germ granule. And so this germ granule is a liquid-like structure where we find proteins like this structural protein PIGL1. And also our PB protein that binds to the piRNA. And so what the piRNA does, it is then transported out of the germ granule, it goes into the nucleus, and where it binds to a nascent transcript, and then the transcript will be degraded. So in the worm, C. elegans that we use, the germ granule that I mentioned is located in germ cells, which are again basically sperm and egg, which are developing in these worms together. And they are these in this structure. They are these green granules that locate to the germ cells, and they are beautiful dots that are surrounding each germ cell nucleus. And they are always present throughout development, when the worms are very little to when the worms are growing up as an adult, and also when the worms are an embryo, they are always present. And so when we look at real scientific images, like microscopy data, we can also identify germ granules by looking at the structural protein PIGL1 that surrounds the DNA, here marked in blue, around the nucleus, and they are very beautiful. So we were interested in, since our PB protein is also located in germ cells, how, if there was any interaction with germ cells and piRNAs. And so again, so we were looking at wild type worms, where we looked at the beautiful structure, and then we looked at PB mutants. So just to point this out again, PB mutants are one of our mortal germline mutants, which become sterile after a couple of generations. So when we look at late generation, when the worms are not sterile yet, we can also see these beautiful germ granule structures. However, when the worms are sterile, we actually see that a lot of them are missing, and the germlines are not as beautiful as they are in wild type. And so we were wondering, is the loss of piRNA and the loss of the PV protein maybe responsible for this loss of germ granules, which is a sterility, which makes them sterile. And so we were asking this question: is the absence of germ granules implicated in the sterility effect of piRNA loss? And in order to look at that, we were looking for some genetic interaction of how these proteins interact. And for that, we were looking at their germline structure. So you have to, this is just very simple slide actually.
Recording stopped.
All you need to know is that the germline in fertile
Recording in progress.
And fertile wild type worms is large and beautiful with a lot of germ cells. But when the when the worms are sterile, these germline structures become very small, and only a few germ cells are present. And so our worms are very sad because they can't reproduce. And so coming back to our two little proteins that we are looking at, when in the absence of the PV protein and in the absence of piRNAs, so worms are still fertile at earlier generation, which is generation 10, for example. And they're also fertile at the same generation in the absence of the PIGL1 protein. However, when we mutated both proteins together, then we could see that most of them would become sterile already in the third generation. And at the same time, when we look at their germlines, we could see that in the single mutant, the germlines would be very large, while in the double mutants, the germlines would be very small, and only a few germ cells would be present, if at all. And this indicated to us that the piRNAs and the germ granule structure were actually interacting together and were maybe responsible for this sterility effect. And so to summarize, we found this important interaction of the PV protein and therefore piRNAs and PIGL1, which represents our germ granule structure. And so what we think here as a mechanism is that the loss of piRNAs might weaken the germ granule structure over time, or that PV and PIGL1 are both important for germ granule structure. And so if you want to know more about this specific effect, I want to point to this publication that I published last year in Nature Communications, where we described this effect in detail. And with that, I want to thank my lab for their support, and especially my PI, Craig Mello, and also my PI from my previous lab, Sean Ahmed, at the University of North Carolina, who initiated this project with me. And if you have any questions, I would love to hear them. So.
Thank you so much, Maya. That was a beautiful work. So if anyone of you have questions, please type in the chat box or raise your hands. I think I have a question from your talk. So you were, I'm curious to know that you said the piRNAs, they can either make a protein and degrade a protein.
Right.
Yeah, so I wanted to know, like, in what conditions and, they make a protein, because contrary to what Milky, said, that piRNAs are always kind of like involved in degrading a transposon. Yeah, so just wanted to, curious.
Yeah, so as Milky said, so transposons are like the type of transcripts that the cells don't want to have, because they can insert into other genes, so they are like bad transcripts. So the piRNAs will target them, and if the piRNAs are not supposed to target the transcripts, at least in worms, there are other small interfering RNA molecules that have a slightly different structure that will also bind to the same transcript. together with the piRNAs. And so then depending of the quantity with the other small siRNAs and the piRNAs together, whichever molecule is like the dominating molecule, it will either be up for degradation or it will be up for translation. Does that make sense? So it's kind of a balance of percentage of how many piRNAs are present on the transcript.
Okay, yeah, got it. Thanks.
Thanks, Maya. That was really nice, and we really enjoyed the data as well. It was very beautiful images. I wanted to ask you, so how does this relate? This was in worms. Does the same mechanism work in humans as well? Is that related to fertility in humans?
Yeah, it's similar. So the piRNA production is slightly different, but that was not the title of the talk. piRNAs are also present in people, and in fact, what I was reading about is that a lot of men have a mutation in the PV protein, and that is what a lot of infertility comes from. So I do not know if there's any treatment for this right now, but I could imagine that in the future there could be some treatment to inject, for example, a functioning PV protein that would then make these men fertile again.
Interesting. Thank you.
I think I have one more question, Maya. So in what conditions there's like piRNAs or PV proteins, they are like mutates? Do we know in humans, are there in specific conditions? Either temperature fluctuations in testes or something like that? Do we know of the causes?
So the PV protein is a protein, so that means in order to make this malfunctioning protein, the DNA was mutated. So it's just from what I was reading, it is just a condition in a lot of men for some reason. It is not the piRNAs that are mutated. Maybe they are, but this is not what I was reading about. So the piRNAs are usually transcribed from specific loci that are piRNA specific loci in the genome. And then the PV protein binds to these piRNAs and guides them to their target. If the PV protein is not there, the piRNA does not know where to go, and it will just be lost. So it is therefore it's not. If you delete the PV protein, the piRNAs are not there. They will just be degraded.
Okay. Thank you so much. Yeah.
Thank you. That's great. We have a question here. So are there different isoforms of PV proteins, or is there only one version? And are they conserved and common to all animals, like the piRNA?
Yeah, so that's a good question. So in worms, there's only one PV protein, which is called PRG1. And in other animals, I think in mice, I don't want to say anything wrong, but the Milky might probably know better than me. I think there's at least 3 PV proteins that have similar functions.
That's interesting.
And so every species has a different number of PV proteins.
Sure, yeah. Is this related to age? Do you see a decrease in amount of PV expression with age?
I study C. elegans. I don't know for people. We definitely see a decrease in piRNA function with age.
Okay.
But we have not published that yet.
Interesting. That's very fascinating.
So I have a follow-up question. So I heard that over time, the sperm counts is going down in males. Is that related to the piRNAs, or is that a separate issue?
Honestly, I do not know the answer to your question. I don't want to say anything wrong. I mean, there's a possibility, but there in advanced age, there's so many things that can happen. Likely, from what I remember, it might be also a lot of DNA damage and.
Right. Okay, thank you. Does anyone else have any questions? Again, feel free to unmute or type it in the chat box. I'm just trying to see if anyone has raised their hand. And I don't see anyone. Okay. All right. So with that, we will wrap up. So I'm just going to share my screen again. Okay. So again, thank you, Maya, and thank you, Milky, for your talks. This was really great. And we very much appreciate you joining in today and presenting. And we want to thank everyone for attending tonight and for asking questions. And so we would also like to ask your help with our future events. So if you could please scan the QR code on the screen and take the event survey. And feel free to suggest other topics that you would like to see in our Science Cafes. And the event survey, I believe, should also be posted in the chat if you cannot scan it from your screen. And again, please join our social media pages to stay up to date with our events. And finally, please look out for an announcement in your email inbox about our next Science Cafe, which will be released in a few weeks. So until then, stay safe, enjoy the fall colors, and we hope to see you again soon at our next event. Thank you all.